All right, so here is our lecture that you need to listen to prior to our class on Monday. And this chapter, chapter 4 from the textbook, addresses the concept of epigenetics. And this is basically everything that impacts inheritance and impacts the particular phenotype beyond the level of the sequence of nucleotides. So there are a number of different factors involved there. Because epigenetics is so big, we tend to focus on things that impact inheritance and the expression of genes, but still are physically associated with the genome. Because technically, various dietary implications, unless they're physically adjusting the particular expression of genes by physically changing something on the string of nucleotides. I mean, those are still epigenetic factors, but they're way too big to try to address in a course on genetics. Talking about epigenetics, but epigenetics is probably the fastest growing field within genetics, and it is quite fascinating. So let's talk about some key terms. First key term: DNA methylation. Probably the most well understood epigenetic feature is DNA methylation. And so there are enzymes that will add methyl groups, typically to cytosine residues, and we'll see this in when we get into the slide. And what it does is it changes the expression of the gene that includes that cytosine residue. DNA methylation tends to lower the level of transcription of that gene. The target of most of our DNA methylations are what are called Cpg dinucleotides, and this is where you have a cytosine residue that is immediately followed by a guanine residue. And I'm not talking about cytosine residues that are forming hydrogen bonds with a guanine residue on the other strand, because that's going to be every cytosine residue, but the cytosine residues are the cytosine nucleotides that are immediately followed by a guanine as the next nucleotide on the same strand. Or another way to say this is a cytosine deoxyribonucleotide that is bonded through a phosphodiester bond with a guanine deoxyribonucleotide. All right, histone modification. Other than DNA methylation, this is probably the next best understood and studied of the epigenetic factors. And here you're not adding methyl groups to the actual bases. Instead, you are adding methyl groups or acetyl groups or phosphates or ubiquitin to the histones. And remember when we were talking about histones that histones are proteins, therefore they're made up of amino acids, and the very beginning of a polypeptide is called the N-terminus because the end of the amino acid on that has a free amino end, and the other end, the end of the amino acid is, or the end of the polypeptide is going to be the C-terminus, where the last amino acid has a free carboxylic acid end. And in the histones, when those polypeptides fold to form the histones, they tend to have their N-terminus sticking out like a tail. And this is where you tend to modify histones. And again, histone modifications usually result in changes in the level of expression. Now here you tend to have bigger changes, like when you're forming heterochromatin or maintaining euchromatin, it usually has to do with histone modification. Now, something to note, histone modifications can overcome DNA methylation patterns. So where your DNA methylation pattern may say don't transcribe this, if your histone is modified in such a way that it says do transcribe this, it can override what your methylation pattern is saying. They can also override other epigenetic factors as well, like what we call genetic imprinting. All right, so we've talked many times about the dosage issue that you have in mammals, where females have 2 X chromosomes and males only have 1, and so one of those X chromosomes needs to be deactivated. And so the way that works is by basically there's a gene called the X-cyst gene that codes for an RNA that does not get translated. And so this gene is the XS gene. Again, it codes for this big, bulky RNA that doesn't get translated and instead blankets the X chromosome that it was transcribed from and triggers other changes that convert that X chromosome into a bar body, convert it into heterochromatin. Genomic imprinting. So one thing that we'll see, and I didn't mention this already, is the methylation patterns and the histone modification patterns. They will be passed on mitotically. So that is, it's one of the major things that leads to differentiation among cells, where you take a stem cell and you shut off certain genes by either methylating cytosine residues or by methylating histones. And so you shut off those sections of chromosomes and shut down those genes. And then you may amplify others by acetylating the histones. And what you do here is you basically lead to different levels of expression of different proteins, which then will differentiate the cells. Now, the DNA methylation and the histone modification are passed down mitotically. So if you generate, say, not a pluripotent stem cell that can become anything, but maybe a stem cell that can become any type of blood cell, every time that new now hematopoietic stem cell that can become red blood cells or white blood cells, every time it replicates to make two hematopoietic stem cells, it passes down its epigenetic programming so that it basically is locked into becoming a type of blood cell. And then if it goes into becoming, a cell that's actively mitotic, say an epithelial cell, at the base of the epidermis, and it's actively mitotic, again, it'll pass down the methylation patterns and the histone modifications mitotically. But these are not passed down meiotically. Meiosis basically resets all of the epigenetic factors for almost the entire chromosome except, and we'll see this in a few slides, the portions in which there's what we call genomic imprinting, where basically the paternal version of that chromosome will have a very specific methylation pattern that the maternal version of that chromosome doesn't have, has a different methylation pattern. And so what it does is it tells the cell where that gene came from. Because if you just look at the sequence of bases, there's no way to tell whether it came from the father or the mother. But the cell sometimes needs to know based on how this gene is going to be expressed. And so the one that came from the father has a specific methylation pattern. The one that came from the mother has a different methylation pattern. And so we call that genomic imprinting. It's very few genes. Again, we'll talk about that in a couple of slides. So the conflict of parental interests is a hypothesis to explain why genomic imprinting exists. And basically the hypothesis states this, and this is something we'll talk a lot about on Monday. The hypothesis says this: the father is most successful when his offspring grow as quickly as possible to reach sexual maturity, right? The father is going to maximize his reproductive fitness by forming as many offspring as possible that reach sexual maturity. The mother's best interest is to, because at least for mammals, they're investing a lot in their offspring. They don't want them to grow too quickly. They want them to grow more slowly so that they can more efficiently feed them while also not taking away from their own nutritional needs. And so the mother's best interest is to actually keep the offspring from growing as quickly as possible, whereas the father's best interest is to get them to grow as quickly as possible, produce as many as possible. And so because of those rivaled interests, the idea is that the mother has a benefit to basically mark her genes that are going to lead to cell cycling in such a way that it doesn't go too quickly. Mark those oncogenes in such a way that that expression isn't too quickly. Whereas the father is like, man, let's just activate all these oncogenes and get the cell cycle happening as quickly as possible. So again, it's just a hypothesis. There are some data that support it. There are other data even within this chapter that we'll talk about in this lecture and on Monday that don't support this hypothesis. Epigenetic reprogramming. So I mentioned up above that the DNA methylation and the histone modification patterns, that they change. They change with when you go through meiosis. They basically reset. And so what that's called is epigenetic reprogramming. So again, mitotically, they are maintained, and every cell that is derived from one of these now differentiated cells with its particular epigenetic pattern is now going to have that same epigenetic pattern. But again, when you go through meiosis, you need to start that over, because you need to produce a new you need to produce a zygote that is pluripotent, and that can then produce other cells that are also pluripotent that can then differentiate in the fetus. And so that's what epigenetic reprogramming is. Transgenerational epigenesis. This is the idea that even outside of the area of imprinting, that there are some means by which you can pass on methylation patterns. And we basically see two aspects of this that we'll talk about in a later slide. All right. So DNA methylation. There are two types of enzymes that methylate, again, typically cytosine residues. One of them is this one, DNMT1. So it's DNA methyltransferase 1 and then DNA methyltransferase 3. This one basically rebuilds the methyl pattern that was in the previous slide, methylation pattern in the previous cell. So again, these are passed on mitotically. So this DNMT1, after the mitotic division, and you have now two daughter cells, DNMT1 will go in and put the methyl groups on the same residues that they were on in the parent cell before mitosis. DNMT3 adds the new methyl groups. So throughout your life, you will methylate various sections of your genome. This one methylates new sites. So these are where we talk about nature versus nurture, and the reality is it's both. It's what's in your programming, but then also your life experiences change what happens as a result of that. So as you go through your own life, or that now daughter cell goes through its own experiences, it will accumulate methylation patterns that are new, and those are put on by DNMT3. But again, DNMT1, DNA methyltransferase 1, rebuilds the methylation pattern that was in the parental cell. DNMT3, or DNA methyltransferase 3, methylates new sites. We sometimes call this the de novo methyltransferase. And so ultimately, the outcome of DNA methylation is the shutdown of transcription. All right, histone modification. Histone modifications, we get what's called the histone code. So you can acetylate these free tails. You can methylate them. You can phosphorylate them. You can ubiquitinate them, add a ubiquitin group, and ultimately this is providing a code beyond just the level of the DNA. And so we've got 4 core histones, and then the whole histone is an octamer made up of 2 versions of histone 2A, 2 versions of histone 2B, 2 versions of histone 3, 2 versions of histone 4, and the N-terminal tails stick out, and we modify them primarily by adding our group to lysine residue, sometimes to an arginine residue, and so you can see the notation for these, the single letter notation for these residues, because then when we have this code down here, this is telling us that on histone 3, on the 9th lysine residue, it is trimethylated. On this one is saying on the 3rd histone at the 1st lysine residue, or no, sorry, the 14th, I saw that as a 1, not a 14. On the 14th lysine residue, it is monoacetylated. And this one on histone 4 at the 20th lysine residue, it is monomethylated. And again, so now we get a histone code that, again, like DNA methylation, is passed down mitotically, but is reset through meiosis. And so these are carried out by enzymes. Histone methyltransferase attaches methyl groups to these histones. Histone acetyltransferase attaches acetyl group, and then you've got histones deacetylases that can remove acetyl groups from these histones if expression now needs to be turned off after it had been activated, if you have to go from euchromatin over to heterochromatin. So X inactivation, this again, it's the inactivation of one of those. We've got a dosage issue in mammals. Females have 2 X chromosomes, and one of them needs to be inactivated. This is random. As far as in an early cell, which one gets inactivated is random, but every cell that descends from that inherits the particular X chromosome that is deactivated. And this is where you could get calico cats, where you get big regions of the body expressing the orange pigment because the particular X chromosome that it got inactivated contained the normal eumelanin, that brown pigment, and the one that is not inactivated contains the altered pigment. Okay, and again, we've got the XIC, the X inactivation center, which is a composite of 7 proteins and 5 RNA products, and almost all of them are transcribed from this area here, the X inactive specific transcript. And this is, again, it's on the X chromosome, codes for non-coding RNA products, these 5 RNA products, and they are reverse complements. They go and they blanket that X chromosome and ultimately deactivate it. All right, so genomic imprinting. So again, I told you most of the time these imprinting sections are shut down when you go through meiosis and are not passed on to the next generation. But an exception to that is with genomic imprinting. So there are some genes, about 100 in humans, that receive epigenetic features that basically are passed down, that they are inherited. And so if they're paternally inherited, the epigenetic programming is different than when they're maternally inherited. Now, most of these genes are on chromosomes 11 and 15 in humans. In mice, we do a lot of our genetic studies on mice. Most of their imprinted genes are on chromosome 7, just a difference in our genomes. But what's cool here, or maybe not cool, what's interesting here is that you can have particular diseases that result if you lack the imprinted code from both parents. So for example, we've got a section on chromosome 15, on the long arm of chromosome 15 from the 11th to the 13th band, where if you only have the maternal imprinting, it's called Prader-Willi syndrome. And so there are a couple ways you can get that. One is if you just completely lack the paternal chromosome and you only have one copy of the maternal. The other way, or at least of that section of the chromosome, if you were missing the entire paternal chromosome 15, you would not be viable. But if you were just missing that portion of chromosome 15, that's one way it can happen. Another way that it can happen is if you have what's called uniparental disomy. We talked about that in our lecture for Wednesday, in which basically when you fix the issue of having a trisomy in chromosome 15, you might actually erase the paternal chromosome, leaving only the maternal chromosome. So that's Prader-Willi syndrome. Some of the pathologies of that syndrome are large size, small hands, mental retardation. In the same region, if you only have the paternal version of that factor, you get what's called Angelman syndrome. And it has some different pathologies, but again, it's it's the lack of having both of those parental copies. So we have some questions. How would this develop? And again, we've got the hypothesis that because the two sexes have different goals or what maximizes their fitness, there are certain genes that they have to mark to ensure that they are expressed properly. And so we'll talk about this more on Monday. And then how does this get reset? So genetic imprinting, because you have to think about it this way. Let's say your dad, the chromosome he gave you of chromosome 15 came primarily from his mom. Okay, right? So that happens. His particular chromosomes during meiosis, you're either going to get predominantly your paternal grandmother's chromosome 15 or predominantly your paternal grandfather's chromosome 15. Well, so your dad's version of that chromosome 15 that you inherited is going to have the maternal imprinting patterns. But then when he passes it down to you, it's going to be passed down with the paternal epigenetic imprinting factors. And so you get kind of an interesting phenomenon in which these imprinting patterns have to be reset. And so you have to remove. Now, most of these imprinting patterns are carried out by DNA methylation of particular cytosine residues. And so you need what are called demethylases that will go in and remove the methyl groups, and then other methyltransferases that will build a new pattern. All right, so here's this epigenetic reprogramming fairly quickly. So here's, by the time you get to the blastocyst stage, you have basically locked in your methylation patterns that are going to be inherited throughout your lifetime. You've set in a lot of the differentiation has happened. You've set cells to become a certain type of cell, either a certain type of blood cell or a certain type of other cell. But anyways, you basically have set that process in place. Now, and that's set in place very early on. But as you start to make gametes, you have to erase basically all of the imprinting that you had. You have to, if you're female and you've deactivated one of your X chromosomes, or you have Klinefelter syndrome and you are a male with two X chromosomes, you have to deactivate one of those X chromosomes. And then that way, so that your offspring can then deactivate their new one. And so that, again, these are carried out primarily by demethylases that will remove methyl groups, and which will then allow the next generation to set it themselves. All right, transgenerational epigenesis. Again, the idea that you can inherit some of these methylation patterns from your parents, even beyond just imprinting. And so here's a good example. We've got mice with the agouti coloration and mice with the yellow coloration, also larger size, tend to be obese and just be a lot bigger. And this, we're actually going to talk a lot about this on Monday. So most of this discussion we'll leave for Monday. But this sounds a lot like Lamarck's ideas on evolution. So Lamarck's ideas on evolution were basically you, during your lifetime, you develop features that then you're going to pass on to your offspring. And so, people that refuted Lamarck's ideas, came up with just like absurd examples where, you'd have a person that becomes a bodybuilder, and they're not going to have offspring that are, stronger at birth than what a typical person would. And so that was kind of an absurd example. And so a lot of Lamarck's ideas were just completely discredited and did not make a big impact on the biological community. And so Lamarck predates Darwin, and so because of Lamarck's ideas on how evolution might work, biological evolution was largely discredited until Darwin and Alfred Russel Wallace came up with a better mechanism, natural selection. But what we find is that there are some epigenetic programs that can be inherited through imprinting or through other mechanisms, and we'll see this especially with this color. So we've got a mutant allele, which is yellow, that if it is methylated is not expressed, and you get the agouti coloration and normal size, but if it is unmethylated, you get the enormous size and the yellow coloration. On top of that, if your mom consumes, materials in her diet while you are in utero that are particularly good at facilitating methylation, it can impact methylation patterns. And so again, we'll see that especially with this specific coloration, this specific version of transgenerational epigenesis. So as you prepare for class on Monday, go over this lecture. You don't need to worry about going through the questions from the end of chapter 4 just yet. We will do those on Friday. And if you have any questions, we'll start our class on Monday addressing those questions. If not, we'll go into some more ideas under epigenetics. All right.